So with 10 panelists and half hour, I don't think there's a ton of time that we have to rile up the speakers and see what we can get out of the various levels of description that we heard of. But I think there's also very little doubt now that the range of ways in which we refer to internal models is pretty wide, going from the single cell levels in cerebellum-like structures, right at the very one end, to it's interesting. It seems like almost people group themselves according to the no, not quite. There's a little bit of a mix-up. It's not going from all molecular to all cognitive, but at the other end is being really probabilistic models. So I'm hoping that all of you will participate and sort of ask questions, but I'll start it with sort of a simple question, which is with the variety of descriptions that were given, maybe each of you can give 3-4 minute synthesis of what you think actually is common among the things that you study versus the other end, especially the people who study neural circuit motifs. Do you think there is anything you study that actually bears on the types of probabilistic models that, for example, Dan and Josh talked about for higher level cognition, and vice versa for people who study more cognitive levels? Where do you see a connection between the neural circuit motifs? Can you imagine that there is actually a linkage to describe what you study based on those neural structures?
Okay. Right, so I talked about negative images in this fish, and there's a lot of parallels between negative images in a fish and motor adaptation paradigms and cerebellar dependent motor adaptation. So the fish is learning something, he's put off target, the neurons adapt, and then you turn the stimulus off and you see a negative image. That's like reaching to a target, you're perturbed, you hit the target, then there's after effects. I understand that in my system by adjusting synaptic weights. I got some representations in granule cells. I've got some weights. I've got a supervised signal from the world that is a sensory prediction error. That's what I understand. But I'm very interested in understanding how to link that to Bayesian ideas, just to take one example. So that's the kind of thing I'm interested in. How do I go from the internal model that I understand at the level of cells and circuits in this, and we could talk more generally about the cerebellum too, where we all of us in the cerebellum are influenced by these Marr-Albus models that are traditional models that have a lot of synaptic weights and plasticity. But where is what I'm interested in is in the connection between those domains and some of these higher level ideas, and I don't know where. I'd be very interested to know and hear what the other speakers think about how to bridge some of these gaps, and what are some of the experiments we could do, or what are some of the mechanisms that we think could help to bridge those gaps. Do the mechanisms look the same as the ones we know? Is it some of the same things but deeper and more? Is it if you have a bunch of these internal models based on synaptic plasticity and some microzones of the cerebellum, and you wire them all up together, could you get some of these hierarchies or some of these other ideas coming out of them, or do we need entirely new mechanisms? So what should we be looking for in the brain, I guess, is one of the things I'm interested in. In our fish work, I think generalization is a critical issue, and that, I think, kind of bears on some of these issues, whether what fish is learning is just a giant lookup table, or how well can it generalize to new contexts. That seems like a critical question about learning and the power of these systems. I talked about granule cells. One of the things you wonder is, when do you start to run out of granule cells? That would be a big criticism of these traditional Marr-Albus models, is you're basically learning some giant lookup table, and what do you do when that runs out? So what are some solutions to these problems, I think, are interesting questions. So no answers, but I think it's good to have the conversation about these kind of things. And the cerebellum is involved in internal models. We know a huge amount about the circuitry of it, and yet there's a giant gaping chasm between what people are doing at the circuit level in the cerebellum and any of these interesting ideas. So it's kind of I think it's an important pressing issue to think about how you might bridge those gaps, because if we can't do it there, I don't know where we're going to be able to do it.
So I can turn this right around. So your model seems very nice in that the model you study is very nice in that it seems like what Daniel talked about structural learning, what I talked about the assumptions that are kind of ingrained in the internal model seem like ingrained in the basis set you found in your granular cells and the distribution of that basis set, because that's going to dictate how learning generalizes, how you make sense of some ambiguous error signals and how these are projected into it. So now it would be interesting to think about would the fish be able to learn a new structure in a way, right? Would it be able to somehow modify its basis set and then learn more arbitrary things that might not encounter in nature, but might, once you expose it long enough in the laboratory, that it actually could acquire this structure? Or ask otherwise, how does this structure come about in the granular cell layers? Is it genetically inborn, or is it something that just reflects the learned structure, the learned prior that it experienced in the environment?
So I'm going to go home and put some yellow and red blocks on my split bell treadmill. We'll see if you can make predictions about what happens. No, seriously, I think that one of the big, certainly from my point of view, one of the big problems that I have is when you use devices or robots to try to train human behavior, the problem of generalization is huge if you want it to generalize. And turning off and on generalization is something that we'd like to be able to do in a better way. And so, when can you pull something into a certain structure, and when can you not? Can you take it out? So I think that that's an area that is very useful to me. Now, the other thing that I would say is in patient groups, we have patients that have different kinds of diseases that affect different cell types. And we don't understand at all how those patients are different. They can look a little bit different, but we don't understand in terms of their learning ability and their ability to control movement, what we would expect to see. So on my end, I mean, I'm way out on the disease end, so I think that these are some of the interesting questions that I would have after this.
Blocks, no, not the blocks. I'm actually going to go for history. So I think it was really interesting watching all the speakers at this meeting and going from the very reductionist to the higher level topics. It's actually kind of an old thing. So 65 years ago, in 1950, two seminal papers were published by Roger Sperry and by von Holst and Mittelstadt, talking about corollary discharge and efference copy, respectively. And 15 years after that, Hans Lucas Teuber founded the Brain and Cognitive Sciences department, which was psychology, but it turned into BCS. And I wasn't around back then. That was before my time. 10 years later, I was born. But from what I understand, back then, there was a lot of talk about this. So corollary discharge from the reductionist level up through Hans Lucas Teuber talking about it at the psychiatric level and talking about lesion studies in humans and the deficits that would occur and how this could be related to Roger Sperry's ideas and von Holst and Mittelstadt's ideas. But at the time, there wasn't any neural evidence for any of this. So Roger Sperry and von Holst and Mittelstadt did really interesting behavioral manipulations, turning eyes around and reversing crossing pathways and things like that, where they could infer a lot. And nowadays we have the neural data, and in humans we have imaging data, and we have really beautiful, elegant psychological experiments. And I'd like to see kind of a renaissance of that integration of the two levels. And I think that meetings like this, I think that we're kind of getting there. I know in my own experiments we've been looking at our perceptive stability of the visual scene across eye movements. And we've been looking at it in human subjects recently, doing psychophysics and asking questions about priors. So, for example, if we make an eye movement and whether or not we notice something moves across that eye movement, I think has a lot to do with our expectation of should it move or not. So if we see a rock move, that would be a lot more surprising than if we see an insect move, for example. Same basic image projected onto the retina, but our priors will influence our sensitivity to that movement. And we have preliminary psychophysics data suggesting that this is actually true. And it'd be really interesting to see and to study that at the neural level and to coordinate with people doing psychological experiments with priors, and like Josh was talking about in the last experiment with kind of physics models of the world, to kind of bring it all together and understand how the brain does that at the neural level and at the psychological level.
Yeah, so I guess for me, it's been a very interesting day. I came from more of a sensory coding, sensory motor transformation background, so I kind of came into this area of internal models, as I said initially, accidentally, and I think it's made very clear to me that when you think about sensory processing or sensory motor transformations, it implies some sort of linearity, which doesn't really exist, because if you think about sensory processing, typically we're sensing the stimuli we sense around us to do things, right? So you're really sensing for action. And I think this is something that, students don't really appreciate until they get towards the end of their neuroscience, because it's something that's evolved later. And in the way we even teach neuroscience, we really focus on this sort of linear representation of sensory processing and decisions and then motor output, but it's really it's a loop. And so I think that's one thing that I've really learned to appreciate. And in order to understand that, you have to have internal models. And I've, again, based on the background of the vestibular system, focused on this simple sensory motor transformation, which is either vestibulo-ocular reflex or vestibular spinal reflexes. But I think what helped, and well, I guess I could say one thing, in listening to Nate's beautiful work, I'm always very frustrated that I can't get to that level in the monkey, and I think about how I can do that, and I talk to Nate, and then I get kind of frustrated, but it's okay. But I guess, in terms of higher level perceptual things that were discussed today, you could take a simple system like the vestibular system, and we're actually starting to record in thalamus, and we see actually the same things, and that's not surprising. We see reafferent suppression. My guess, if we did one of these training paradigms, we'd see that as well. So this opens the question of how distributed is the suppression or the internal models that we're looking at, and we all assume very often they're occurring in the cerebellum, and there's evidence to suggest that some parts of them are, but then we see that it's more complex, right? And I think that's something that I don't have a good handle on there are some types of internal models that are probably in the cerebellum, but it's, what is an internal model? And, these are things that are open questions. And sort of the fact that we see the reafferent suppression at the level of thalamus, it's similar to what people are now seeing in visual cortex and auditory cortex. So it's a very general principle, which I think it's really in the last several years that we appreciate how fundamental action is to the way we process. So I think it's actually a really exciting time in that way.
Great. So I guess I'd like to just contrast Kathy's and Nate's to Josh and mine. So I think we think of probabilistic models, and the reason we think of them as probabilistic models, but I think you don't think of them as probabilistic maybe, is that it's really important not only to have an answer, but have knowledge about the certainty in your answer. And that's because often you want to take that answer and combine it with another answer, and you know how, not just what the answer is, but how reliable it is, or combine it with a cost. And so we're very interested, I guess, in not only the answer, I'm sure in the blocks task, if you said to people, is it going to fall over or not, and how confident are you about that, people will give you very accurate answers on really how reliable their answers are, and we find that in our tasks. So I'm curious to know whether you think, it sounds like you think it's a deterministic thing, you just get an answer, but not that you represent your certainty in that answer, or do you think these circuits also know how certain they are about the object out there? Because that's going to decide what to do about it. And the certainty really plays an important role, or if you want to then take the head signal, combine it with vision or something, you need to know how certain these signals are. Do you see those things, and do you think you're doing this probabilistic computation going on really in that sense? Is it like to go backwards?
Somewhere I think not in my structure.
You think not?
Is going to give the best answer it can, and then forward that to some
higher stage that needs to take that into account.
Okay.
So it's going to make a decision.
So the variability in the neural firing, and there's nothing there, and there's no signal in that or something which passes on to say,
No.
I think there's something out there, but I'm not sure. Compared to, I'm absolutely certain from the signal there's.
Mm-hmm.
That seems. Then that's an impoverished version of the Bayesian version, maybe to some extent.
No, I guess. I don't know. In many systems, including in our system, even at the first stage, you get feedback from the highest stages after a few synapses. So you certainly couldn't rule out that those synapses are coming back, that information is coming back, and we haven't explicitly looked for it. So it could be.
Yeah, I guess it's just, I mean, I guess one of the things we see in motor behavior, right, is there's variability. And I mean, one of the things we'd like to do, the question we have is if you start to apply perturbations just to kind of keep you in the noise band or poke you out, what's the tolerance? So I don't know if that gets at your question, but there probably is some level of tolerance. I mean, I don't know how much precision we can get to answer your question, right? Because there is, every time you see a little bit of difference, but my guess is there'll be some nonlinear point, some point where suddenly you're in a different regime, right?
So several people during the course of the day have admitted that they were in domains that were beyond their comfort zone. And I think that is important in trying to answer Mehrdad's question or any question in neuroscience at the moment, that you find yourself in a region where you feel inadequate and stretched. And so as a circuit empiricist, the reason I'm sitting next to the guy on the left of me is not an accident there, because my role in life for however long is scientifically to provide circuit explanations for the generic synthesis that Daniel and people like Daniel provide, where they pose questions from engineering, biomechanical perspective that need understanding in a different currency, the currency of neurons and circuits and behavioral output functions. And how long will it take to achieve that merger? I don't know. Some things, I think, in the course of a decade, and when I listen to Josh, right, I'm a little more pessimistic about coming up with causality explanations of what you're describing, or Rebecca or someone like, because these are very different. These are the things that are the topic of the next 50, 60, 100 years to try and provide. But you have you have to start somewhere, and you have to start with the fact that there are 86 billion neurons in the human brain, and it's not an accident the way they're connected. And that is what makes this, to me, an exciting endeavor to try and, then there are the enthusiast, and how far that leap of, interpolation is going to be, I think, is an individual topic for, some people. I remember when Rod McKinnon was at a meeting and he announced a dramatic change in his research program on potassium channels to move from, one subunit and gating to something that was on the same potassium channels, 5 angstroms away in the intracellular domain. For Rob, that was the leap of a lifetime. For us, it's somewhat less. And so I think what we've heard today is Mehrdad was very clever choosing this topic, because as many people have commented, it means different things to different people and gives you the substrate for different. I'm at one end of that continuum, and I want to get to the other end, and I know I won't. And that's we don't even know how many nerve cells there are, different classes of nerve cells in the spinal cord. And the stuff that we have been doing suggests that there are 200 different classes of interneurons that are controlling motor output. So if that is true, you realize immediately that the whole thing is hopeless, and you have a lot to do, and you're never going to be out of a job.
You're not nearsighted like me, Tom. That's the problem. But the 30,000-foot view, I mean, I'm quite heartened by seeing parallels between mammalian and bird systems. I mean, it's really just I'm going to speak specifically about our own work, but to peer into premotor networks and see an architecture that connects to the auditory system, I think that was surprising to me. But the generative aspects, this is, again, I approach it pretty much one cell at a time, but generative aspects in birdsong are really an important part of learning, and that's something I'm curious about, both in terms of statistics, but also in terms of how much those individual and generative processes interact with the model. A beautiful aspect of birdsong is you can implant at least one part of the model in the bird's brain by just playing it a sound. But it seems like it has an ability to generate its own improvisatory behavior on top of that that ultimately lends itself an individual song. Even though it's very similar to the one it copies, it allows it to be a little bit different. And so there's a hint of individuation and self-determination in that process that I think is quite profound. I'll leave it at that.
So my general feeling about trying to link up the two extremes is that in terms of looking at neurons and trying to understand circuit mechanisms, we need to come up with some sort of, underlying principle that can explain all the individual components that we're seeing, right? And so, the analogy that I have in my head is like you have, a video of a bouncing ball, right? You can monitor every pixel in that video, right, and try to understand what is going on in that video. And you can do that, and it's likely to be sort of an inefficient representation. But if you could somehow figure out that there's Newton's laws going on, right, then you have a really concise summary of many individual components, all of which are changing over time. And so if we can come up with something like that, right, from at the circuit level, right, then that might be the link point to these higher level concepts that Daniel and Josh are talking about. So sort of looking at the work that I presented this morning, I think this is sort of in the middle of the two ends of the spectrum. And so I'm going to try to relate to each of the two extremes. So I think the relationship between the work that I presented and sort of the higher level sort of Bayesian view of things is that we really view this internal model that we're extracting as a subject's prior assumptions about the world, right? And so where we're going with this is to try to use these concepts to understand learning, right? And then trying to link to sort of lower level circuit mechanisms. I think it would be really interesting to address these shortcomings that I mentioned in my talk, which is that we're not actually recording the corollary discharge or from the neurons that are actually implementing these internal models. And if one day we could do that, then trying to incorporate that into this internal model formulation and trying to figure out what is the structure of the internal model that can best explain what we're seeing in the neural activity in these experiments.
I'm not sure where to begin except to say just the idea of 50 to 60 years to bridge cognition and neural circuits, again, I would call that optimism and not pessimism. And I think that's exciting. I think I might even still be alive then. When you read Craik from 1945, it sounds like an impossible gap, right? Or when you look at a lot of cognitive psychology, and you don't even know where to begin to understand how all the 86 billion neurons or the 200 types just in one small part of the nervous system relate to the cognitive level. But I guess part of why I'm excited about this whole spectrum of what you have here, and again, compliments to Mehrdad for putting together a really interesting group that it raises these challenges, but actually I think offers hope and optimism, is that there are some aspects of human cognition that are really the core, the heart of the matter. They're present in young infants. There are ways in which even a 12-month-old is smarter than any machine, system, or robot that's ever been built. So it's really about what makes us smart. And yet it's these things that do relate just to what Byron was saying. It's basically some kind of computational approximation to Newton's laws on objects that can make sense of this common sense physics that the infant has somehow internalized, right? And again, I'm optimistic not only in the connections that people here, but the work I showed briefly at the end, Jason Fisher's work in Nancy Kamwisher's lab, right? That the different kinds of tasks we're studying, there seem to be parts of the brain that are specialized for doing these kind of computations. It's very preliminary, so we'll see how that plays out. But let's say they're right? Again, this is way out of my comfort zone, but I don't think it's an interesting, maybe suspicious coincidence that one of the two main places that they're finding a selective physics signal is in the premotor area, right? The idea that there's a coupling between the internal models in the motor system that we use for planning our actions, that were the topic of most of what the other speakers were talking about, and that you can study in all sorts of ways in neural systems in all sorts of different species, and that there might be a direct link between those circuits and the ones that are at the heart of human common sense cognition about the world. That's very exciting. And I don't know what the other speakers think of that idea. If there might be anything there between, maybe another way to put it is Mehrdad titled the symposium "Internal Models in Sensory Motor and Cognitive Function," and I think most people were talking about sensory motor function as a cognitive scientist. It seems to me, and Daniel and I were maybe two of the people who were talking about cognition. But if we look at the part of cognition that it might share something functionally, computationally, with the sensory motor systems, which have these internal models which you can study in all sorts of very powerful neural circuit ways, then maybe there's a neural circuit commonality too that might have some hope of answering the kind of thing that Tom is looking for.
Maybe we should open up also to the audience. I was trying to think myself whether there was some sort of qualitative divide between the sensory motor and the cognitive, and what would that divide be? And I don't think it, well, I'm going to just say that everyone here was talking about learning, models based on learning, and there either was or certainly could be a probabilistic aspect to all of those learning models. So I don't think that was it. Whether animals have access to knowledge of their probabilities, I'm not quite sure. But at least in terms of the mechanisms, they're just probabilistic learning mechanisms leading to something you could call a model. But it would seem to me that may be a qualitative difference are the difference between models that are strictly feed-forward based on learned priors about sensory input and then expected outcomes, and the models that require actual simulation, sort of what you were talking about, like with the premotor. So it's one thing to build even like a generative face model, which is very complex, but actually, the way you talked about it, it could be done without all kinds of iteration, recursion, and so on. Just strictly feed forward, straight through the kinds of things that neuroscientists, sort of know how to study. But then when you imagine, like, well, what is it like to imagine shaking the table and simulating the blocks falling off, which I guess half the audience, solve the problem that way. That is not, that's involving all kinds of iteration, recursion, and so on and so on. That to me seems qualitatively different when you get into those kinds of physics models. What happens if I get in my car and I get into traffic, and how am I going to get to work? That kind of planning, and that may be the step that's beyond what us neuroscientists normally can make progress with.
So I want to be a little bit more optimistic there. So I think premotor areas is a really interesting zone to be in, because, for example, from Richard's talk, we saw that in the equivalent, in premotor equivalent in Songbird, there seemed to be quite a high-level temporal representation of motor behavior that is being extracted in HVC. We see in our work that we teach people a range of different fine motor skills, and premotor cortex looks like it extracts some regularity out of this pool of motor skills that it needs to learn, and kind of lifts it to a more abstract level that it can then flexibly recombine. So there seems to be something about the circuit of premotor cortex that is really good at not just learning by rote learning or repetition, but somehow discover in the neural network those regularities and then utilize them in new situations. This is, of course, much more modest than what Josh talked about, but you could see how those circuits might, something about these circuits might be then extrapolated to solve something that is more akin to cognition. So I think there are some interesting links here that I think might be valuable to follow through.
Wait, can I just quickly follow up on also just one thing in answering Bob's question? So, yeah, I guess I think you're exactly right that that's a very interesting distinction, but I think it's not just human cognition that has that ability to do some kinds of mental simulation, right? I mean, I think I came from a very interesting conference last week the FENS Brain Conference in Denmark, and there were a bunch of people, for example, Lauren Frank, another great alum of our program, studying various kinds of these hippocampal ripples. He came out of Matt Wilson's lab, and again, locally we've seen this from Matt and other people working there. So that's there are places where some kind of forward simulation planning algorithms that you can study in rodents, might plausibly be doing something similar. Howard Eichenbaum had similar ideas in other kinds of not just spatial aspects of hippocampal function. So I think you were exactly right to look at that, and I think they're going to be very different kinds of neural processes that will do these different kinds of inference in internal models. It's just it's not only humans that do the interesting kinds of planning.
I just wanted to ask the panel if what is the opinion of you in terms of connecting the two the two extremes of the spectrum, meaning psychophysics and measuring behavior by humans of relevant tasks on one hand, and then the other end, which is poking electrodes and looking into behavior of neurons or even neuroimaging. So how should we bridge the two opposites?
Well, in my view, this is the basic problem. It doesn't matter whether one is, you want to extend, as I mentioned before, and I think that in the motor control system, the insights that have emerged from human motor physiology serve as an example of what should be driving experimentation at this neuron physiology circuit level. And you see many, many examples of that in this vestibular system world, or this world of, different strategies of motor activation. What you can do in humans, albeit slightly, associative, is amazing, actually. And many, many models have emerged from human physiology that I think are not being paid sufficient attention to from the animal experimental group, partly because these worlds don't collide often enough. So to me, the whole purpose of being in neuroscience is to extend yourself beyond something that you think about a lot and take a big leap and see what you can do to bridge that distant gap. And how distant, as I was saying, is the subject of individual choice.
But can you extrapolate from sensory motor to, for example, face recognition or speech recognition, language comprehension?
Not easily at the moment, but the things that Daniel was talking about and the way in which decision evaluation has an impact on some sensory world is telling you that there are things that you need to understand that we don't understand at the moment. And I mean, there are a lot of those things, but you could say that any two of the talks that you've heard today, there are points of contact which could be driven forward. So Mark talking about, colliculus interfrontal eye field is a set of problems that could be analyzed. There's very little work, just to sort of bring it down to earth, on collicular function using modern molecular genetic approaches, but there are people in Cambridge who are beginning to try and look at that. So I think the optimistic thing is that nothing is beyond the range of pursuit at the moment. It's just that there are a lot of things that you could turn your attention to.
I think also I'd like to emphasize the worth and importance of basic research, and pretty much everybody on this panel does basic research. We're studying the problems. We don't know what the answer is going to be. There's not a definitive disease that we're trying to cure. Amy is probably the one exception here who's actually trying to help people. We're trying to help people, but in a more abstract way, I think. There are a lot of animal models that can be appropriately chosen that can get give us a lot of insight into human behavior with the right preparations that are used, the right behavioral tasks that are used. Rich's work in birdsong, for example, looking at the neural circuits, looking at all the way from the neurotransmitter level with a lot of anatomy, but getting into vocalization and learning and communication between conspecifics that has direct analogs to, I think, human behavior in a lot of ways. A lot of basic research has been done on invertebrates, which aren't really represented here. Beautiful corollary discharge work in crickets and other invertebrates and insects. But that has really informed my work in primates and I think the work in humans as well. So it's a beautiful spectrum, I think, across the animal kingdom that we really need to support this basic research to get at these fundamental questions.
I'll just add that we are big consumers of the animal research, and we pay very close attention to what kinds of timescales of learning we can expect in different structures in the brain. Understanding how in animal work that people dissect the different kinds of learning signals that you need to drive different systems. So, you know, it's turtle scratch. I mean, we read turtle scratch because we want to understand spinal networks. So it's not that we aren't consumers of those things. And I agree that if you don't do that kind of work, I don't think that you're going to advance any of the types of work that I do, for example.
Thank you for your talks today. I'm from a very different background, so my background is mostly about the biophysics of the network. And I'm trying to put together the group of talks that I heard today. So, you know, one thing was common was the internal model across all these talks. And some talked about that how, like when they're evaluating the internal model, they're trying to basically see how something could compensate for the errors that are basically created. But there's something to be said that is the nervous system is always behind the world, right? Like you can never have it lag zero at the beginning of your experiments. So how do you want to deal with that situation? Because no matter where you start and how you start, you're starting with a system that already is making predictions or trying to basically compensate for the lag time that it goes through. And and some others actually through the later they said, okay, maybe we have a breakdown of this black box of internal model, and then we have basically a serial of internal models building up on top of each other. And then again, another concept comes into the mind, that is that if you have such thing, then if an error occurs in the system, right, then how does the system tries to compensate? Like if you have such sort of like serial categorization of these internal models, then how do you want it to compensate for the real world or what's happening on the outside? And that brings basically, I think that the connection to the last series of talks is that I think what's the last two talks were mostly about the higher functions. And there, everybody talks about how you have like some probabilistic model of your world, and maybe you know there's an overarching principle here that you can go through. But there's something to be said that is that it's not just the brain that can act in this sort of manner, right? But you have any physiological system. Let's say your parasympathetic system is also composed of excitable membrane. It has many common elements from biophysical principle, and it can compensate for the perturbations that happen in the external world. So maybe what we are trying to do in crossing from basically the biophysics to cognition is something that has to be gone through basically this complex adaptive system format of the network. That this network, as it gets bigger and bigger, of course, it gets to do more complex things. But and then that's how it reaches the cognition, perhaps. I don't know. That's just like a very speculative, like 100 years from now kind of speculation. But I think what's missing here is that no one is talking about like how a complex system replies to the external alternating or altering, let's say, environment from biophysics point of view. And I think that is something that could be investigated and eventually could link all these things together.
Wasn't that what Nate was doing? Yeah.
Well, that's what, but as I said, like, I think there's a misconnection from there to basically the a general principle of how these systems could react to a complex system, in a complex system point of view. That's I think, a difference. But of course, it's the basis of it.
So I just with this question that I think reiterates the complexity of connecting the two ends of this line. I want to thank again all the speakers. It was a fantastic day. Thank you so much for coming, and we will have reception outside. Everybody can sort of continue discussions afterwards. Thank you.